Hello, everyone, and welcome to today's live broadcast, addressing the challenges of bioconjugation for improved cross-linking and modification, presented by Greg Hermanson, Chief Technology Officer and Principal at Aurora Microarray Solutions, Incorporated, and the President of Greg T. Hermanson, Incorporated. I am Xavier Gutierrez of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about a sponsor, visit www.thermofisher.com. Now, before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing your presentation, please use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, if you would please join me in welcoming Mr. Hermanson, I will now turn the presentation over to him.
Thank you, Xavier. And let's go to the second slide and we'll see where we're going today in this seminar. We've got a lot of ground to cover, so we're moving right in. The outline for today's seminar is we're going to look at functional groups on biomolecules, proteins, oligonucleotides, and carbohydrates and glycans. And then we're going to look at some of the principal reactions that are done in bioconjugation on those biomolecules, namely amine reactions, thiol reactions, and carbonyl reactions. And during the course of the seminar, some of the important knowledge that can be gained will include what reactive groups are available and the reactions used for them in bioconjugation, the major products of each reaction, the hydrolysis and side reactions that each one might have, interfering substances that you might want to avoid, how to optimize the reactions, and tips and tricks along the way. So let's jump right into functional groups on biomolecules. Functional targets on biomolecules are really highly diverse. On proteins and peptides, they all contain an abundance of targets for conjugation and immobilization due to the variety of amino acid side chains that are present. And some of the principal ones that are often targeted are amines, thiols, carbohydrates, carboxylates, and glycans. The folding, one thing to keep in mind is that the folding of the polypeptide chain may prevent some reactions from occurring with residues having low accessibility. So not every protein has every residue available. An antibody, for instance, may have dozens and dozens of amines, but maybe 15 to 20 of those amines are readily accessible and highly reactive. It can also target groups on carbohydrates and glycoconjugates. Depending upon the type of carbohydrate, it might contain a reducing end, for example, an aldehyde, or within its structure, it may have adjacent hydroxyl groups or diols that can be oxidized to aldehydes or amines that can be directly targeted, or even carboxylates that can be coupled to. And glycans are present on glycoproteins and glycolipids, and we'' look at their structures in some detail in a few slides. On DNA and RNA and nucleotides, reactions can be targeted at the 5' phosphate group, the ribose diol at the 3' end on RNA, and certain positions on the bases. Nucleophiles on nucleic acids, however, are often less reactive than nucleophiles on proteins. So there's some specialized reactions that have been developed for oligonucleotides that we'll look at in a moment. So if you looked at reactivity in proteins, this is an example peptide that just happens to contain just about every functional group that can be targeted or modified in a protein molecule. And if you look from the left side towards the right, on the left-hand side we have the amine terminus, And we can target amines on the alpha amine or on the lysine epsilon amine side chain, the third amino acid over. We can also target the carboxylates on aspartic acid and glutamic acid. A very popular target are thiols on cysteine residues, the residue right in the middle. Others that can be modified, however, include the guanidinium group on the side chain of arginine, the phenolic ring on the side chain of tyrosine, and even tryptophan can be modified with some specialized reagents. Methionine is also reactive, but it's rarely targeted specifically because some of the products of those reactions are actually reversible and labile. So in targeting methionine, you also target cysteine, but the methionine, any reactions with methionine will break back down. And finally, you have the imidazole ring on the side chain of histidine, which can be iodinated or reacted with alkyl halides. Around the outer perimeter in red of this group of peptides are the principal reactions that can be used with each of these functional groups. With oligonucleotides, it's a bit different. Whether it's RNA or DNA, you can target the 5' phosphate group if it's present. And that's usually targeted much the same way as a carboxylic acid can be activated with the water-soluble carbodiimide EDC. And with oligonucleotides, you do that in the presence of imidazole, and that forms an activated phosphoryl imidazolide, which can be then reacted with amines, hydroxides, or Aminooxy groups to target and couple to specifically only one end of the oligonucleotide. With RNA also, the 3' end can be targeted through pyridate oxidation of the diol on the ribose at the 3' end. And after oxidation to form aldehydes, they can be coupled with amines, hydroxides, or Aminooxy groups as well. The base groups, if you notice that guanine, cytosine, and adenine all contain what appear to be amines. However, the amines are rarely reactive because they're actually in resonance with the ring structure, which dramatically decreases the reactivity of these NH2 groups. But ring structures the carbon atoms on some of the rings, like guanine and cytosine in particular. Guanine can be brominated with N-bromosuccinimide and coupled to amines through the bromination point. And with cytosine, bisulfite can be used to activate one of the positions on the unsaturated carbon-carbon bond. And that leads to the ability to do a transamination reaction at the NH2 group with substitution of a spacer arm that can be then coupled to using other reagents. So the reactions with oligonucleotides are a bit different than proteins, but the chemistry that's been developed has been well developed and works very well. With functional groups in glycans and carbohydrates, these are often modifiers of protein molecules or lipid molecules, and they have complex structures, but within the structures, there's certain points that can be pyridate oxidized, either diols on, for instance, beta-D galactose, or on the diols on terminal sialic acid residues. In dilute solutions of pyridate, the diols on sialic acid can be targeted specifically at close to 0 degrees C and will leave untouched the beta-D galactose residues. At higher concentrations of pyridate and room temperature, you can oxidize also any beta-D gal residues. So once forming the aldehydes on the glycan, you can couple with amines, hydroxides, or Aminooxy groups also. So let's look at the major reactions done with these functional groups. First, of all the dozens and dozens of reactions that have been developed for bioconjugation, there's probably about a dozen. I've listed 10 here that are commonly used and repeatedly used and available on many of the reagents that are sold commercially. That's reactive esters, which include NHS esters, TFP esters, or NHS carbonates, which react with amines to form amide bonds. You can also activate a carboxylic acid using a carbodiimide, like EDC, and reacted in the presence of NHS or sulfo-NHS to form an NHS ester or sulfo-NHS ester. And that activation product can then go on to react with amines to form amide bonds. And we'll look at this reaction in great detail in a few slides. Malaimide groups are very popular to use. They're available on many cross-linking reagents or modification reagents, and they react with cysteine thiols to form thioether bonds. Aldehydes can be produced, as we saw with glycans and carbohydrates. They form shift bases with amines, and if they're reduced with reductive alkylation, they form secondary or tertiary amine bonds. Or they can react with hydrazide groups to form hydrazone bonds, or form oxime bonds with Aminooxy groups. So there's a variety of options with aldehydes. Pyridate oxidation is often used as an intermediate step in the reactions of bioconjugation to form aldehydes from diols, used to create reactive sites, especially on carbohydrates and glycans. And disulfide reduction is often used. This is the formation of thiols for conjugation from disulfides within proteins. And this uses DTT or TSEP or other thiol-reducing agents. Epoxy groups react with amines, thiols, or hydroxyls and form secondary amine, thioether, or ether bonds. And epoxies are often used to immobilize ligands onto particles and chromatography supports. Haloacetyl groups like iodoacetyl or bromoacetyl reacts with thiols rapidly to form thioether bonds. And also photoreactive groups can be used for nonspecific targeting when studying protein-protein interactions, for example, and there's a variety of photoreactive groups available. And cycloaddition reactions have gained popularity, especially the click chemistry reaction with copper or copper-free methods to form a cycloaddition product. But in looking at even these most popular reactions, there's three sets of reactions that occur most often. And let's look at amine reactions. With amine reactions, the most popular reactive group in existence right now is the N-hydroxysuccinimide or NHS ester. It is certainly essential technology for amine conjugation, and it's used in many dozens and dozens of reagents available to either cross-link molecules or modify them. An NHS ester is the most common reactive group used in bioconjugation, and they are spontaneously reactive with amines and almost always work well. There's two principal types of NHS esters, a standard NHS ester or a sulfo-NHS ester. And if you look at the compounds below, on the left-hand side, it shows two compounds that use NHS esters. If you look on the left-hand side of those compounds, you'll see the NHS ring structure, which is esterified to a carboxylic acid. And on the right-hand side, the two structures show the same compounds, only with sulfo-NHS groups. And the presence of the sulfo adds a negative charge character to the entire compound, which is strong enough to water solubilize the compound prior to conjugation. So NHS ester reactions with amines involve a acylation reaction with the amine, where the amine group, unshared pair of electrons being highly negatively charged, interacts and is attracted to the carbonyl carbon atom, which is on the NHS ester because that's partially positively charged. This interaction causes collisions, and it eventually kicks off the leaving group, NHS, to form an amide bond. And the amide bond is highly stable. And that's why NHS esters are popular. This reaction proceeds rapidly, and the product of the reaction, the amide bond, forms very quickly and is stable. The resultant bond is stable, and the reaction occurs slightly at alkaline pH in aqueous buffers, or an organic solution can be done with a base present, like DMAP or TEA. The optimal pH and conditions are dependent, however, on the type of NHS ester compound being used and the rate of hydrolysis that may occur at any given pH. Typically, we say that NHS esters or sulfo NHS esters work best between pH 7 and 9, but it's really important to optimize for each different compound that's being used. Some of them will work best around pH 7 or 7.5. Others may work best at pH 8.5 or 8.7. It just depends upon the type of compound that the NHS ester is present upon. Now, reactions with NHS esters with amino acids and proteins occur with the amines principally, which is the N-terminal amine, alpha amine, and the lysine epsilon amino groups to form the amide bond. And that's the major target. So if you're reacting an NHS ester containing crosslinker with a protein molecule, you're anticipating that that is going to result in an amide bond, mainly with lysine side chains and with alpha amino groups. However, it can also react with histidine through its imidazole nitrogen side chain to form a carbonyl imidazolide. Now, unfortunately, this product is unstable and in itself reactive. It's a reactive intermediate that unfortunately reacts very quickly with water and hydrolyzes. So if there is a lot of histidine present in a protein, you may actually need to optimize the amount of NHS ester containing reagent up a little bit in concentration to overcome the accelerated hydrolysis that histidine might cause in this type of reaction. NHS esters also can react with serine or threonine hydroxyls to form esters, but it's often difficult to get this reaction to specifically occur in aqueous solution. The same with tyrosine OH groups and cysteine sulfhydryl groups. These are side reactions that sometimes occur. Fortunately, cysteine doesn't often occur in free form in most proteins, so when an NHS ester is reacted with a protein, cysteines are not the principal target. And with serine and threonine hydroxyls or tyrosine hydroxyls, we'll look at those next because this is an interesting side reaction that's been understood only recently. So NHS esters normally have difficulty modifying serine hydroxyls in aqueous solution because of the competing effect of a much higher concentration of water. But when serine OH groups happen to be held in proximity to amino acid side chains having a transient modification potential, then the hydroxyls can be modified. And an example of this is either in a single peptide or in a series of polypeptide chains where a histidine side chain imidazole group is held in proximity to a serine hydroxyl group. The NHS ester actually can react with the histidine imidazole group to form this unstable carbonyl imidazolide seen in the middle. And because the OH group is close by, an NO shift, acyl shift can occur and transfer the NHS ester containing compound over to the hydroxyl on serine. This can be seen with certain proteins and certain peptides, and it's been studied by Madler et al. In 2009. Now, the same type of reaction can also occur with tyrosine and threonine hydroxyls, where if the histidine occurs in proximity to either a tyrosine OH group or a threonine OH group, the same type of a reaction can occur with initial complexation with the histidine nitrogen and then an NO acyl shift over to the corresponding OH group. The relative reactivity of this potential reaction is serine occurs more commonly than tyrosine and tyrosine more commonly than threonine. But it does occur. So if you're modifying peptides and you happen to have histidine groups near serine, threonine, or tyrosine residues, and by mass spec or other means you find that some of those OH groups have been modified, this is indeed probably the side reaction that has occurred. NHS esters also have a competing hydrolysis reaction, which is unavoidable in aqueous solution. In general, hydrolysis occurs as the pH becomes more alkaline. Hydrolysis occurs with increasing rate. Alkalinity probably increases the nucleophilicity of the unshared pairs of electrons on the oxygen and water because there are fewer protons available to associate with them. So NHS esters typically are most stable at pH values between 3 and 6. So if you are working with an NHS ester and need to purify an intermediate in aqueous solution, having it on the acid side will give you a little bit more time to purify it with limited hydrolysis. In a paper published at the beginning of affinity chromatography and the reactions to immobilize affinity ligands on the chromatography supports, Quattrocrissaz and Parikh studied NHS esters and their coupling of amine-containing ligands to agarose, and they looked at the rate of hydrolysis at pH 6.3 and 8.6. And as you can see here, at pH 6.3, the rate of hydrolysis is considerably less than the rate of hydrolysis at pH 8.6. So if you're reacting a molecule at elevated pH, up around 8.5 to 9, you may have unacceptably rapid hydrolysis, which will compete with the actual coupling reaction. Similarly, with sulfo NHS esters, in 91, Grumbach and V studied sulfo NHS-LC biotin to see what the relative rate of stability of the sulfo NHS group on the compound would be to changes in pH. As you can see, around pH 3 or 4, the half-life of hydrolysis is hundreds of minutes in length, but as you climb up to the areas where the reaction would actually be done, the half-life at pH, let's say 7.2, is less than 50 minutes. At pH 8, that drops to around 10 minutes. At over pH 9, you're down to just a few minutes of half-life as the rate of hydrolysis quickly accelerates. Now, an alternative TFP ester to NHS ester is the TFP ester. That's tetrafluorophenyl esters. They react also with amines to form amide bonds, similar to NHS esters. TFP esters, however, have been found to provide better hydrolytic stability and higher yields of amide bond formation than NHS esters, especially at alkaline pH. So some reagents are available now with TFP esters on them and NHS esters. And you may want to try both types of reagents if you're optimizing a bioconjugation procedure, because TFP esters, not only are they more stable to hydrolysis, but they have better yields of amide bond formation. In the two graphs at the bottom, and the one on the left studies the reaction to hydrolysis at pH 7 of a TFP ester compared to an NHS ester. And you can see after 240 minutes, about 20% of the TFP ester was hydrolyzed, whereas at the same time frame, 50% of the NHS ester was hydrolyzed. So you get better hydrolytic stability of a TFP ester and better amine coupling capability. And even at pH 10, which is extraordinarily alkaline for hydrolysis of NHS esters, the NHS ester was essentially completely hydrolyzed after about 150 to 200 minutes, whereas TFP esters still had 60% of their activity left even after 240 minutes. Some of the interfering substances to be aware of for NHS ester conjugations are listed here. Primarily nitrogenous buffers containing primary or secondary amines, for example, tris or imidazole. Beware of amino acids and their derivatives, such as alpha amino groups on gly, cis, arg, and hist, which are often added to buffers. Also, glutathione contains a primary amine. So if you've got a cell extract and you haven't gotten rid of small molecules, glutathione is present in high concentrations and may be a primary interfering agent with NHS or TFP esters. Secondary and primary amine containing polymers, for instance, polyethylene imine will react with NHS esters. Also amino sugars, glucosamine, or hydrazide, hydrazine, or Aminooxy compounds. Ammonium salts too. Now, ammonium bicarbonate, ammonium citrate, ammonium acetate, ammonium sulfate, etc. are often additives to buffers to stabilize or extract proteins from cells or tissues. If the ammonium ion isn't removed and it's present in high enough concentration, it may compete in the reaction. Now, fortunately, ammonium is almost always protonated in aqueous solution, but there is an equilibrium, and NH3 will also be always be present to a certain degree. And it's NH3 that's going to react with either TFP or NHS esters. So it's better to remove ammonium compounds before doing the reaction. Azide, too, often added as a preservative in buffer solutions, reacts to form acyl azides. It's a moderate nucleophile, and it will interfere. Some people have done successfully conjugations in the presence of 0.02% sodium azide, but I would recommend typically avoiding azide and removing it prior to conjugation reaction. Be careful of old urea. It may break down to carbamic acid, which then decomposes into CO2 and ammonia, the ammonia being the interfering substance. Also, thiol-containing reducing agents, DTT, 2-mercaptoethanol, 2-mercaptoethylamine, and other thiol-containing compounds. The thiol will react with an ester, an activated ester. And beware of other proteins or blocking agents added to a solution, which may contain amines, such as BSA. They're often added to antibodies. So if you purchase an antibody and you want to conjugate or modify it, make sure it doesn't have a carrier, BSA or something else that contains an amine. Another way to form a reactive ester is the use of water-soluble carbodiimides. And carbodiimides like EDC or EDAC can be used to form reactive esters from carboxylates directly in aqueous solution. EDC is called a zero-length crosslinker, which means it forms an amide bond between a carboxylate-containing molecule and an amine-containing molecule with no intervening linker arm. And it's frequently used to couple antibodies or other amine-containing molecules to carboxylated particles, especially magnetic particles. And this is the typical reaction that you'll see, that EDC can react with a carboxylate to form an intermediate O-acyl isourea derivative. So the intermediate ester is the component that actually reacts with the amine to form the final product, the amide bond. However, this typical reaction sequence isn't describing really what goes on, and EDC does have some potential deficiencies. And I don't want to go into great detail in this slide, but if you look at EDC, the top compound in the center, in order for it to be reactive with carboxylic acid, it needs to be first protonated. That intermediate protonated form can hydrolyze at that point and hydrolyze to an isourea, which is then inactivated. It can go on to react with a carboxylic acid to form the O-acyl isourea derivative, and that derivative needs to be protonated again to form an intermediate reactive carbocation. And that reactive carbocation can also hydrolyze back to the carboxylic acid and the isourea product, essentially inactivating it at that step. If it goes on to react with the amine, it gives the desired product, the Amide Bond Formation, which is over on the right-hand side. But even that reactive carbocation can then cyclize to form an anhydride, or if EDC is used in too high of an excess, you can have an acyl shift to form an acyl isourea derivative, which is then inactive. So you can see that EDC reactions are not quite as simple as the previous slide and many of the reaction sequences in the literature describes. There's a lot of places where EDC can have side reactions towards hydrolysis or competing reactions that deter or diminish the amide bond formation. There was a paper in the mid-90s that studied EDC in each of the reaction steps that I showed in the previous slide. First, with regards to hydrolytic stability of EDC versus pH, they found that EDC is quite stable from about pH 6.5 on up above pH 9, almost pH 10. And you can see all of those points indicate that the activity remaining even after 300 minutes is nearly 100% at higher pH values. Even slightly on the acidic side, it was still very stable. However, dropping down to acidic values at 3.95 or 2.52, it was rapid hydrolysis of EDC at those two pH values. So keep this in mind because they also studied the rate of reaction of EDC with carboxylates versus pH. And in this case, it was found that the best pHs for activating carboxylic acids were on the acid side from about 3.5 to about 4.5, with activity also being possible at higher pH values. So at the pHs where EDC hydrolyzes the fastest, it's also the most effective at reacting with carboxylic acids and forming the intermediate ester. Now, what about amide bond formation using EDC? Well, it turns out that amide bond formation, if it's done in a one-step reaction where everything's in solution simultaneously, and if you look at the graph on the right-hand side, ethylene diamine and in the yellow dots was present in the solution with the carboxylate containing compound and EDC simultaneously, and these represent the yields of amide bond formation in a one-step procedure. If a two-step reaction was done where the carboxylic acid was reacted first, activated, excess EDC was removed, and then ethylene diamine was added, it decreased the yield somewhat, but even at higher pHs, it remained stable in terms of the yield of amide bond formation. With benzylamine being an aromatic arylamine, you would expect the reactivity of an arylamine to be somewhat less, and their yields of amide bond formation in this experiment were indeed less than ethylene diamine in a two-step. In reality, using EDC reactions in this type of procedure, whether one-step or two-step, a pH of about 6 provides very good compromise between the rate of hydrolysis of EDC. EDC is quite stable at pH 6 in aqueous solution, and the yield of amide bond formation is at a maximum or nearly a maximum at pH 6. However, you still get the potential of all those side reactions that EDC could have. So how do you make EDC reactions more efficient? It's possible actually to limit EDC's side reactions through an initial reaction with NHS or sulfo-NHS. So in this case, we're creating an intermediate NHS or sulfo-NHS ester that will provide better stability towards hydrolysis and won't suffer from any of the EDC rearrangement reactions. So this reaction occurs if you have this showing a particle with carboxylase groups on it, reacting with EDC to form the intermediate oacyl isourea reactive ester. And then with sulfo-NHS in solution in excess, this reacts with the EDC ester to form the sulfo-NHS ester intermediate, which then goes on to react with the amine-containing compound and form the amide bond. Staros in 1986 published a paper which demonstrated that in the presence of sulfo-NHS, the EDC reaction was dramatically improved and the yields could be very high compared with the same reaction done without sulfo-NHS present. So let's move now to thiol reactions, particularly Malaimide reactions with thiols. Malimide reactions are actually the second most popular bioconjugation reaction done, behind only reactive ester coupling to amines, which we just looked at. Thiols are also the second most popular targeted functional group behind amines. So that's why you find thousands of literature references that cite the use of Malimide reactions in creating bioconjugates and dozens of commercially available bioconjugate reagents, which have one or more Malimide groups. They are often reacted second in heterobifunctional cross-linking procedures because they usually are more stable than the reactive esters in aqueous solution. So in a crosslinker containing a reactive ester plus a Malimide, the reactive ester would be reacted with an amine, let's say on a protein first, and then the secondary reaction would occur, which is shown at the bottom here, where the Malimide ring with the double bond would be reacted with a thiol containing compound or peptide, and the thiol would couple to by addition to the double bond to form the thioether product. However, this reaction doesn't really tell the full story. This is a typical reaction you see in the literature, but what really can occur is shown on the next slide. This is some of the true reactions that occur with Malimide groups. They are more complex than most schemes would lead one to believe. The desired product is accompanied by hydrolysis products and a number of different derivatives, including the potential to create different stereochemical species. So if we look at this reaction scheme, the Malimide on the upper left, initially it can react with the thiol and form the thioether product as we saw in the previous slide. The Malimide ring also can hydrolyze in the presence of water to open up the ring structure to a malamic acid derivative, which is shown just below the Malimide. Now, the ring opening can occur on either side of the ring and form two different stereochemical products. Unfortunately, both of these hydrolysis products are relatively unreactive with thiols. So the hydrolysis of Malimides essentially inactivates the double bond to the addition of thiols. However, if you do successfully link a thiol-containing molecule and form the thioether derivatives shown on the upper right, notice that there's two derivatives that are possible. And even though they look similar, they're actually two different stereochemical forms. And so if you're working with a research conjugate, it doesn't really matter because you're probably forming a conjugate that's going to be used in a detection procedure and it won't be used in vivo. However, if you're working with therapeutics, forming antibody-drug conjugates or something else, these stereochemical products can make a difference in activity in vivo. Now, once the thioether product is formed, the ring structure of the malamic acid that's left can also hydrolyze. And I'm showing two of those hydrolysis products, but if you recognize that I'm only showing the ring opening on one side of each of these, there's two other products also that can form. So from one successful thiol reaction with a malamide, you can get two different thioether derivatives and four different hydrolysis products. So there is a potential that you could have 6 different products in solution simultaneously. Malimide reaction rate also was studied with sulfo-SMCC modified proteins. So BSA was activated with sulfo-SMCC, which contains an NHS ester on one side, sulfo-NHS ester, and a malamide on the other end. And BSA then gets modified with sulfo-SMCC through an amide bond, and the excess malamide on the other end of the crosslinker sticks off of BSA, can be used to create immunogens from thiol-containing peptides or other molecules. So cysteine was coupled in this experiment to malamide BSA for the indicated times in the graph, and then the amount of cysteine left in solution was immediately determined using the Ullmann's assay. The difference between the amount of cysteine initially in the reaction and the amount remaining in solution at any given time equals the amount coupled to the malamide groups. So what you can see from the graph is that the Malimide reaction is essentially complete within 2 hours, and usually a lot faster than that. And this is at pH 7.2. And if you're working with proteins that are activated with sulfo SMCC, I would let them go to 2 hours because you're going to approach 100% coupling yield at that time frame. Now, an early paper by Gregory, the box over on the right-hand side, in 1955 studied the stability of N-ethyl Malimide and its reaction with sulfhydryl groups in solution. And what they found was with a variety of sulfhydryl groups, N-ethyl Malimide could couple with them in solution, small molecules, within only a couple minutes getting nearly 100% yield. So the slower reaction with proteins is simply due to the fact that the protein and the Malimide modification on the protein is present at much lower concentrations than what Gregory analyzed back in 1955, but still the Malimide thiol reaction is very rapid. So even with the number of side reactions and different products that can form with this reactive group, it is still used in research to a large extent and diagnostics. The only time you want to be careful with it is what will happen when you're making a therapeutic compound by this linkage reaction. Now, Malimide hydrolysis also has been studied. The stability of a Malimide group is dependent not only on the pH, but also upon the neighboring chemical groups. The more alkaline, the greater the hydrolysis rate will be, but different crosslinkers can provide dramatically different stabilities in aqueous solution. And what has been found is crosslinkers like SMCC and sulfo SMCC have relatively high stability at pH 7.2 due to their cyclohexane ring next to the Malimide group, whereas sulfo MBS and sulfo SMPP are much more labile to hydrolysis, as the graph shows. However, if you've modified a protein with one of these Malimide crosslinkers and you want to purify that modified protein away from excess crosslinker, they can be purified by size exclusion chromatography rapidly at pH 6 with little to no loss of Malimide activity, because at pH 6, the Malimide ring only hydrolyzes very, very slowly. Now, there's a couple of caveats to keep in mind. Number one is the reversibility of Malimide thioether bonds, and this has been found recently in a number of publications. The presence of high concentrations of thiols in solution has been shown to potentially reverse the thioether bond. And this is done by undergoing an exchange with the second thiol through a retro-microreaction process, where the thioether on the left-hand side of this reaction scheme slowly can lose that thiol and reform the Malimide double bond. In the presence of another thiol in solution, that second thiol, if it's present in higher concentration, then can substitute for the first thiol on the Malimide ring, thus kicking off the first conjugate and replacing it with a second. Also, a nearby amine group held in close proximity to the Malimide thioether, such as lysine epsilon amino group, has been shown to cause an SN shift, which can effectively cleave the initial thioether bond and form a secondary amine bond. So the thioether in the bottom reaction sequence is neighboring or right close to a lysine side chain amine. If that thioether bond should, through a retromicro process, come off, reform that double bond, the thiol won't be able to find and react again with that double bond before the amine gets to it, and you can actually cleave off part of the conjugate. This reaction sequence actually was found to occur in a Malimide conjugated antibody drug conjugate because albumin actually held the drug in its cleft, human albumin, in vivo. And in the cleft of albumin was a side chain lysine that was held in proximity to the Malimide thioether linkage, and this is exactly what happened in aqueous solution or in serum. What are some of the interfering substances for Malaimide conjugations? The obvious, thiol-containing disulfide reducing agents, avoid them. Cysteine thiol, glutathione too, it's typically at 5 millimolar at least in animal cells. Also avoid TSEP and other strong reducing agents like sodium borohydride. Getz et al. In 1999 observed that when labeling with Malaimide, both TSEP and DTT substantially decreased labeling efficiency compared to no reductant being present. Also be aware that alkaline pH increases hydrolysis and also promotes side reactions with amines. Recently, it's been found that group 6 oxo metalloids such as molybdate and chromate both will quickly hydrolyze maleimide rings and open them. Also, metal salts in samples will rapidly oxidize thiols to disulfides or facilitate that oxidation. For example, metal salts in blood extracts and purified blood proteins, which typically have high iron content. In this case, add EDTA or EGTA to inhibit oxidation of thiols. And finally, BSA. Albumins typically contain a single free thiol group, which will react with Malaimide. So be aware if you're doing a conjugation and you add BSA as a stabilizer to the thiol conjugation reaction, it may in fact conjugate with BSA instead of your intended target. Now finally, let's look at carbonyl reactions, particularly those with aldehydes and ketones. So aldehyde and ketone reactions in bioconjugation are probably the third most popular conjugate reaction that's done. Amines being number 1, thiols being number 2, and reactions with aldehydes and ketones number 3. Now with amine groups, aldehydes and ketones can form shift bases and are unstable and easily hydrolyze. In order to stabilize them, the first reaction shows that the shift base can be reduced with sodium cyanoborohydride to form a secondary amine linkage, which is stable. The second reaction shows the reaction between an aldehyde and a hydrazide group. The hydrazone bond is much more stable than a shift base linkage with an amine and an aldehyde, but I would still recommend reducing with cyanoborohydride because it forms a much more stable substituted hydrazide. The final reaction, an Aminooxy group with an aldehyde, forms an oxime linkage, and this is much more stable to hydrolysis and doesn't typically need to be reduced afterwards. So proteins and amine-containing molecules can be modified to create aldehydes, and in this case, using a reagent that has a TFP ester on one side, a PEG-12 spacer, and then a formyl benzoate molecule on the other. So it has an aryl aldehyde group on one end and an ester, reactive ester, on the other end. It can be reactive with a protein molecule, an amine-containing molecule, and form an amide bond. And the other end of the structure will create aldehydes on the protein surface for further conjugation. Also, aldehydes can be formed from diols and glycans on other carbohydrates, can be oxidized with sodium periodate, as we looked at previously. And sialic acid or beta-D-gal residues within these structures can be oxidized with periodate to form two sites of aldehyde residues. One of the most interesting reactions is the reaction between the Aminooxy group and the aldehyde to form an oxime. This can be actually bioorthogonal if care is taken during the reaction. The reaction is more efficient than reactions between amines or hydrazides and carbonyls, and oxime bonds are actually more stable than hydrazone or shift base linkages. The hydrolytic stability of amines formed from ketones is generally greater than those formed with aldehydes, except for aromatic aldehydes like the crosslinker I showed in the previous slide, which actually forms very stable oximes. No reduction is necessary in the bond to stabilize the oxime, and if aldehydes or ketones are used, cross-reactivity with normal functional groups on biomolecules is avoided due to a lower tendency to form shift-base interactions. So an aryl aldehyde, like in the crosslinker we just saw, will not form shift-base crosslinks with amines in solution, but they will react almost bioorthogonally with an aminooxy group to form an oxime linkage. Now there's one problem with this reaction, and that is with an aldehyde and an aminooxy group, the conjugation reaction occurs rather slowly. And some conjugation reactions are slowed down because they're done at very low concentrations typically. And this can significantly slow down the rate constants. And Oxime Bond Formation, in a paper published recently in 2015, was found to be the slowest of several common bioconjugation reactions. For instance, it occurs considerably slower than a thiol Malaimide coupling reaction. So if we look at the table, the aldehyde aminooxy reaction to form the oxime at pH 7 at 10 millimolar concentration and room temperature, the rate constant was found to be 0.0013, fairly slow. The copper-free click reaction, by contrast, done at only 50 micromolar concentration and room temperature, had a much greater rate constant of 0.9. And the copper-catalyzed click reaction, done at 48 micromolar and room temperature, had a rate constant of 3.4. But I mentioned previously that a thiol coupling to Malaimide was very efficient, and this was done at only 15 micromolar at room temperature. Its rate constant was 734. So you can see the aldehyde aminooxy reaction, while it forms a nice bond and it can be bioorthogonal, in its native state, just with these two reactants present, it's awful slow. So catalysts have been searched for to accelerate this reaction. Years ago, Jenks in '64 identified aniline as a catalyst to accelerate hydrazone formation, and it works quite well for oxime formation as well. At pH 4.5, aniline was found to increase the reaction rate of oxime formation over 400-fold over uncatalyzed reactions. And since that publication, some other catalysts have also been studied, and most of them are either aniline derivatives or diaminobenzene derivatives. Some of them enable the rate of the reaction to be hundreds of times better than aniline alone. And how does this work? So if we look at this type of reaction, you can think of an aminoxy modified molecule reacting with an aryl aldehyde modified molecule at the top. And aniline is put in solution at 15 to 20 millimolar concentration. So it's much higher in concentration than the typical concentration of two proteins reacted together. So the first reaction that occurs is the aniline nitrogen forms a shift-base linkage with the aryl aldehyde group, and that's seen in the middle, intermediate aniline shift base. It just so happens that that aniline shift base forms a better intermediate for the amino group on the aminooxy modified molecule to attack the carbonyl in this linkage and form the final oxime linkage. So the rate of the reaction of forming the oxime is actually catalyzed by the aniline. This works very well, and the other catalysts on the previous page, some of them even work at accelerated rates than aniline does. So in use, they can be used to, for instance, modify a particle with aminooxy groups or aryl aldehyde groups, and then immobilize onto the particle the corresponding aldehyde or aminoxy containing ligand, and forming the terminal oxime bond in the presence of aniline. Works very well. Some interfering substances to look out for aldehyde or ketone conjugations are listed here. There's a lot of biological molecules, especially in cell and tissue extracts, which contain aldehydes or ketones. Aldehyde-containing compounds include reducing sugars and derivatives, glucose, mannose, galactose, ribose, erythritol, maltose 4-phosphate, pyridoxal phosphate, glyceraldehyde 3-phosphate, acetaldehyde. Ketone-containing compounds include keto sugars and derivatives like xylose, acetone, pyruvate, oxaloacetate, dihydroxyacetone phosphate, progesterone, testosterone, cortisol, levulinic acid, and 5-aminolevulinic acid. And the concentration of aldehydes or ketones produced through normal metabolic processes can vary from low nanomolar concentrations to high micromolar concentrations. So they can compete very effectively with aldehyde or ketone modified molecules that you want to conjugate together. Other molecules containing amines can also interfere, especially when conjugating amines on proteins. Buffers containing primary or secondary amines can interfere when conjugating with amines. Also, a caveat is to not rinse glassware with acetone if working with hydrazide or Aminooxy compounds, because any residual acetone will complex with hydrozides or Aminooxys very effectively and essentially inactivate the hydrazide or Aminooxy. Also watch out for formaldehyde, which is often released when oxidizing sugars with pyridate. Now, all of these small molecules that may be potentially interfering, they can be removed using dialysis or size exclusion chromatography prior to the final conjugation reactions. So finally, how do we create an optimal conjugate? We've looked at a lot of the functional groups. We've looked at 3 of the principal reactions that are done on those functional groups. But how to create that optimal conjugate? And there's a number of steps that I think are very important to follow if you want to optimize and produce a possible conjugate. And that is to design the conjugate with the final application in mind as the most important thing to start with. The best performance test in doing this is the actual use of the conjugate in the desired application. Also review the literature for starting points on the reagents, reactions, and conditions to use, knowing that all of those things will have to be optimized further for your application. Use hydrophilic bioconjugation reagents when possible. So there's many bioconjugate reagents, dozens and dozens that are available or have been made. If you can find those that have hydrophilic characteristics and are water soluble, many of them contain discrete polyethylene glycol spacers, DPEG spacers. Also optimize the conjugation method to achieve the highest possible activity for the conjugate. This means to test different concentrations and molar ratios, different buffers and additives, different pHs for the reactions, the temperature being highly controlled. Every potential parameter or factor should be controlled for. Be careful not to include substances that may interfere with the reactions. For example, tris or imidazole in amine reactions. You can remove most of the small molecule interfering substances through size exclusion chromatography or dialysis. Use care to prevent exposure to conditions that may damage the conjugate or its components. For example, light sometimes damages organics, and some conjugation reagents are highly sensitive to light. Oxidants can damage, reducing agents can damage others. Azide can interfere. Use design of experiment methods to test multiple factors simultaneously to achieve the best possible manufacturing process. Finally, optimize the storage conditions to ensure long-term stability and make several batches using a final process to ensure reproducibility. If you take each one of these steps in order and optimize each step, you'll end up at the end with the best possible conjugate for your application. Now, before I close, I'd like to bring up a couple of very important reagents that I think are highly useful for developing conjugates, especially if you're developing conjugates for important applications like in diagnostics or therapeutics. And that is, Thermo has come out with some premium grade reagents which are extra high purity. They have an enhanced level of testing and characterization. They're specially designed for these type of applications with lot retention policies, change management services, and they have very consistent manufacturing with batch-specific manufacturing documentation that can be reviewed. So I would encourage you to, if you're developing critical conjugates for products, to think about using these type of reagents from Thermal. And also, to make it very convenient to do multiple development tests, Thermal has introduced no-weigh reagents, which reflect the fact that you don't have to weigh them out. Very convenient. Pre-weighed dry reagents available in a single-use PCR strip or individual screw cap vial. So there's milligrams quantities of these reagents within the vials, and all you have to do is add a buffer to dissolve them or an organic solvent to dissolve them, and you're ready to go. And this helps save time by avoiding the need to weigh chemicals. Just add water, buffer, or solvent. Helps reduce waste, and you don't have to go into a bulk bottle repeatedly, which can damage the remaining compound within the bottle. Also, I'd like to point out before we close, for additional information, I encourage you to go to the latest edition of my book, Bioconjugate Techniques. Has a long introduction section to bioconjugation describing many, many applications. It provides step-to-step presentations of protocols, which makes the book an ideal source for researchers who may not be familiar with all the reactions and the protocols. It features full-color illustrations, molecular models, and many, many reactions. The new edition also includes an extensive introduction and one of the most thorough overviews of immobilization chemistry ever presented. And you can find this at my website at greghermanson.com or at Thermo Fisher Scientific as well. And finally, just to point out some of the disclosures from Thermo Fisher, the webinar has been provided by Greg Hermanson Incorporated. It's sponsored by Thermo Fisher Scientific, but they don't endorse anything I say. I hope everything has been very useful for your research needs and your bioconjugation needs. And thank you again for attending.
Thank you, Mr. Hermanson, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click on the Send button. We'll answer as many of your questions as we have time for. Now let's get started. Our first question is, seeing that a Malaimide reaction can give so many different products, do you recommend using it?
That's a good question. It certainly gives more products than the traditional reaction scheme originally presented in the literature. And we've come to know more about it over the last 10 years or so. But even with those side reaction products, for most bioconjugation reactions, it works very well, extremely well. It's very specific for thiol conjugation, and it reacts very, very rapidly. So for standard reactions and conjugates used in research and in diagnostics, I would recommend highly using it. The only care I would say is investigate thoroughly the potential for different forms of the final product if the final use of the conjugate is to go into in vivo use of any kind.
Okay, and you mentioned that TFP esters give better yields than NHS esters or sulfo NHS esters. Do you recommend switching to TFP esters to make conjugates?
TFP esters are a great alternative to NHS esters or sulfo NHS esters, and if they are available, I would highly recommend testing both types of esters on the same compound side by side to see if in your application the TFP ester will work the best, because it usually does result in higher yields for the final yield of the conjugate. So yes, I do recommend TFP esters when they are available.
If I form an NHS ester or a sulfo NHS ester on a particle using EDC, can I wash off excess reactants before conjugation without losing too much activity to hydrolysis?
Yeah, that actually is typically done with particles. The two-step EDC sulfo-NHS or NHS activation protocol forms the intermediate activated ester. And if you're at a pH in forming that ester, which is not too alkaline, I wouldn't recommend doing it anywhere above pH 7.5. But you can quickly wash off the excess EDC and NHS or sulfo-NHS from the particles before adding the final molecule to conjugate to them. It works very well.
Excellent. We have time for one more question. What is the best pH to use with NHS ester reactions?
Good question without an exactly definitive answer. Now, I would say the best range of pH is somewhere between pH 7 and 9. But each compound that you're using with an NHS ester needs to be studied for the optimal pH to use. For instance, a water-soluble compound where the NHS ester is not next to any hydrophobic structures usually hydrolyzes faster than an NHS ester on a hydrophobic compound. So if you have a hydrophilic compound, you're probably going to find the optimal pH to be somewhere in the low 7s, 7 to 7.5. If you have a hydrophobic compound with an NHS ester on it, the optimal pH may actually be above 8.0. And with reactive fluorescent dyes with NHS esters on them, a lot of times the optimal pH will be around pH 8.5. But with each new compound, that optimal pH condition should be studied, but most often it's going to be somewhere between pH 7 and 9.
All right, thank you. I would like to once again thank Mr. Hermanson for his presentation. I would also like to thank Thermo Fisher Scientific for making today's educational webcast possible. Now, before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through September of 2019. Please share with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.